Hello and welcome to my talk, a probabilistic programming approach to investigate the coevolution of genes and phenotypes in birds. My name is Viktor Senderov, and I'm a computer scientist. I'm also a postdoctoral researcher in Hélène Morland's lab at École Normale Supérieure in Paris. I work together with biologists and mathematicians, and I use probabilistic programming languages to study the evolutionary history of bird color patterns. These are some of the extraordinary colors of the Estriodid family of finches. This talk will consist of three parts: a brief overview of bird coloration, then what our mathematical model shows so far, and to conclude, a crash course in probabilistic programming languages. The answer to the question, what's the cause of the extraordinary diversity of bird coloration, may depend on whether we're interested in the evolutionary drivers of bird color diversity or whether we're interested in the mechanisms by which bird coloration is formed. The evolutionary drivers have been studied extensively and range from communication and sexual selection to camouflage and predator avoidance. We also understand quite a bit about the developmental mechanisms by which overall bird color is produced. In fact, color can be generated through deposition of pigments or by structural features that interfere with light. For example, the darker color of some populations of rock pocket mice that inhabit dark lava substrates is caused by coding changes in melanocortin 1, MC1R. Our study focuses on the mechanistic understanding of how bird color patterns are produced. A useful analogy is to think of a children's coloring book. We believe that color patterns are evolutionarily more conserved than color itself. For example, a group of related birds may have different colors but still share a similarity in the size and shape of the color domains. But given the apparently overly complex coloration, how do we even make sense of it? It turns out that even though the appearance of birds is very diverse, their color patterns can be systematized in a color map. For example, the two finches that we see here, the zebra finch and the Gouldian finch, may look very different, but their coloration is roughly divided in the same domains, separated by borders of roughly the same shape. So we photographed and measured around 350 bird species representing all major avian families and recorded their color patterns by measuring the number of domains, the shape of the borders between them, and of course the color of each domain, as well as specific characters such as the presence of stripes, patches, etc. Our objective is to apply phylogenomic comparative methods and link this data to genomic sequences using information about the phylogeny of these species. Thus, we want to see which genes are implicated in the evolution of distinct patterns on an evolutionary scale, which will then spur further experimental studies with bird embryos to identify the molecular mechanisms these genes use to form the patterns. Based on previous studies, we compiled a list of candidate genes that may be associated with bird color pattern formation. Some of them are agouti and agouti-related protein, which controls the amount and distribution of eumelanin, brown-black, and pheomelanin, yellow-red pigmentation in the mammalian coat. ALX3, which encodes a nuclear protein that functions as a transcriptional regulator involved in cell type differentiation and development. And EDN3B, which acts upstream of or within melanocyte differentiation and pigment cell development. The next step was to download genetic data for these genes for the 350 or so chosen bird species. In fact, we have made a choice to phenotype only such bird species for which there is publicly available genomic data. We downloaded the protein genomes from NCBI and ran BLASTP with a reference sequence from the domestic chicken Gallus domesticus. Then we repeated the procedure with the coding DNA sequence, but used tBLAST-N instead. This data is then combined with the phenotypic data and placed on the tips of a phylogenetic tree downloaded from birdtree.org. To summarize, our data is a phylogenetic tree, the tips of which have been enriched with categorical variables representing a phenotypic character, e.g. the number of color domains, presence of stripes, etc., and with an aligned nucleic or protein sequence for the genes of interest. Moving on to the analysis part. Many approaches have been proposed to test for association between genes and phenotypes. The veteran of the group is GWAS, Genome-Wide Association Study. It's a regression method from biomedicine and relies on the assumption that the samples are IID. In phylogenetic data, the data points are clearly not IID, as the evolutionary tree represents their degree of relatedness. For this and for reasons such as phenotypic plasticity, genetic linkage, differential selection pressures, etc., models have been proposed that take the evolution process into account. PGLS, phylogenetic generalized least squares, is a regression approach that accounts for the non-independence of the samples. Even though it can be, usually it's not used for testing gene phenotype association, but rather for figuring out the relationship between two traits, say body size and temperature, while controlling for the phylogenetic relationship in the data. While regression can be used for prediction, i.e. given a new predictor value, we can compute a new response value, it's a hard problem as we need to estimate the magnitude of the effect of the predictor. This can further be confounded by the fact that the true relationship between the predictor and the response may be nonlinear and unknown. The DNDS ratio is the ratio of protein changing over protein non-changing mutations on an evolutionary branch. A DNDS of more than 1 suggests that the site is rapidly evolving. To link genotypes to phenotypes using DNDS, we would look for genes that have high DNDS values in the phenotype of interest. However, high DNDS ratio can indicate positive selection or neutral evolution or even negative selection under certain scenarios, making the interpretation sometimes challenging. Another family of methods are profile methods that take into account the direction of selection. In those methods, a fitness profile is used to represent the relative fitness of each amino acid at a given site. An example of a profile method is Pelican. A disadvantage of the profile method is that they operate site by site, the site being a single nucleotide, a codon, or an amino acid. Our method belongs to a group of methods that link the phenotype to the evolutionary rate at which the genotype evolves. In the QT-BERTS algorithm, we model the coevolution of genes and phenotypes directly by introducing 3 rates. Lambda, the rate of phenotypic change, mu, the rate of molecular change, and nu, the rate of joint phenotypic molecular change. In the present version of the algorithm, we assume these rates to be constant on the tree. However, they're unique for each site, or I should say for each gene character pair. We model the evolution of genotypes and phenotypes generatively. The meaning is as follows. Waiting times are sampled from a Poisson process. If a lambda change occurs, only the molecule evolves. If a mu change occurs, only the phenotype evolves. And if a new change occurs, then simultaneously the phenotype and the genotype move. For genes and phenotypes that are linked, we expect to detect higher new values than for genes that are not linked. Note that the individual processes are not constrained to symmetric processes such as Jukes-Cantor or MK. In fact, lambda and mu are the total rates, while the individual transitions may have their own rates. In the example, I've shown that instead of Jukes-Cantor, you can use the Kimura model, which provides for different substitution rates for transitions, so within pyrimidines or within purines, and transversion, so between a pyrimidine and a purine. The total joint transition rate nu is not broken up into further rates, however. So if a simultaneous jump occurs, then the actual transitions that occur are governed by the instantaneous probability matrices obtained by the molecular and the phenotypic process. Another assumption is that the molecular rate does not differ between sites on the same gene, although it can differ between genes. But why not just combine the three rates in one big Q matrix? Here I'm giving a Q matrix for a two-state MK model combined with a four-state Jukes-Cantor model for a single nucleotide. This is an 8 by 8 matrix. However, as the length of the locus increases, the dimensionality of the full Q matrix becomes intractable as the product of the dimensions of the individual sites and the character. For example, for a gene of length 100 and a binary character, the dimension would be 2 to the power of 201. So the model using a combined Q matrix is only applicable on a site by site. But we have to make certain assumptions if we don't want to work with a full Q matrix. This means that we'll process, i.e. compute the likelihood of the nucleotides one by one. It also means we need to simulate the S jumps for each site, as we want to share the information about what happened between the gene and the phenotypic character. Simulation results look promising. Data simulated under the cute birds model under a regime of independent evolution and correlated evolution forms different patterns. Inferring back the evolutionary rates correctly recovers higher nu when correlated evolution occurs. In the next year, we will apply the method on real data. But let's step back for a minute and discuss how we use models to learn about unknown things. Often it's the case that we have a physical system that we would like to study, and even though sometimes we can build the system and study directly, often it's not possible or too costly. That's why we create a model of the system and we do our analysis, reasoning, and design on the model. In our case, the evolutionary past is long gone and we only see shadows of it in the fossil record, in the DNA, and in the present-day phenotypes. So we need to create a model. A probabilistic programming language is a special kind of programming language. A program written in the PPL exactly encodes a statistical probability distribution conditioned on observed data and a given model. Furthermore, using advanced compiler techniques without any extra effort, but writing the generative model, we can sample from it, do model comparison, and infer unobserved parameters. To give you a taste of what PPL looks like in practice, let's take a look at the hello world of probabilistic programming, namely, is a given coin fair? A PPL looks just like any regular language. This one is called WebPeople and happens to look like JavaScript, with the addition of probabilistic constructs. Sample to generate samples from a prior distribution, observe to reweigh or condition the likelihood of the computation on observed data, and infer a higher order function to utilize a number of statistical sampling procedures, here sequential Monte Carlo, to compute the posterior distribution of the hidden parameters under the model conditioned on our data. An important thing to note here is that the user does not encode the SMC. It's provided automatically by the compiler. The coin looks fair according to the posterior, even though it's of a non-existent country imagined by AI. By the way, you can try WebPeople online in a browser. Just search for it online. Finally, in our computations, however, we make use of a specialized phylogenetic probabilistic programming language that we've developed in-house named Tree People. To learn more about Tree People or about bird color pattern evolution, ask me any questions or check out our websites. Thank you so much for your attention.